Hello, everyone, and warm welcome to all of you who have joined us here today. My name is Bruna Lousada, and I'm the European Manager of Teledyne Hanson, and I'm delighted to be hosting this webinar with Dr. Hanan Osman-Ponchet, the founder and CEO of PKDERM. Some of you may remember Hanan from our last webinar in November, in vitro permeation testing application to cosmetic products. And during the Q&A session, there were many questions related to the complex process of conducting an IVPT study. So it has taken us longer than expected, but we have worked hard to prepare this very special webinar for you, which is Mastering in Vitro Permeation Testing, a comprehensive guide to conducting an IVPT study, where we will be addressing most of these questions. So today we'll dive deeper into the topic and cover everything you need to know about conducting an IVPT study, including videos and expert tips, and we hope that you will find it both informative and valuable. So just before we begin, I would like to remind you all that all attendees are currently on mute, but you can submit your questions at any time using the Q&A section at the bottom of your screen. We will do our best to address all of your questions at the conclusion of the presentation, but if we run out of time, we will follow up with you directly via email. If you experience any issues with your audio or connection, we recommend logging off and logging back on. And additionally, this webinar will be available for replay on the Teledyne Hanson YouTube channel in the coming days. So without any further ado, I would like to invite Dr. Hanan to take over the presentation.
Hello, everyone. I am very happy to present the webinar on how to perform IVPT. I hope this webinar will be useful for the audience. My name is Hanan Osman-Ponchet, and today I will show you the different steps of conducting IVPT using excised human skin. You will see during this webinar how to prepare skin samples, how to prepare diffusion cells, and you can see how to make topical application, and finally, and this is the most important step, how to separate stratum corneum, epidermis and dermis at the end of treatment period. So let's go. So IVPT, or in vitro permeation testing, is a critical tool for understanding drug delivery into the various layers of the skin, and it plays an important role in the optimization, comparison, and selection process of drug formulation. IVPT allows to study the permeation of drugs through skin samples in a controlled laboratory setting, and provides insight into how different formulations, delivery systems, and skin types impact drug delivery. IVPT can also be used for bioequivalence studies of topical generics as an EMA and FDA-approved bioweaver alternative to costly clinical studies. In addition to its use in drug development, IVPT is also used for safety assessment of chemicals, agrochemicals, and cosmetic products. What is the objectives of in vitro permeation testing? In fact, as I said, IVPT is a valuable tool for characterization of the rate and extent of transdermal or topical delivery of test items. The main purpose of this testing is to evaluate how the test item is distributed into the skin, including the stratum corneum, epidermis, and dermis.
IVPT is also used to assess whether the test article will penetrate the skin and reach the systemic circulation, and to what extent. This is particularly important because drugs that are intended to be delivered transdermally, as the skin acts as a barrier to prevent the entry of foreign substances into the body. So by measuring the drug's permeation rate and extent, researchers can determine how the drug is distributed in the skin, as well as its potential for systemic absorption. Now, IVPT is typically conducted using a Franz diffusion cell. Franz diffusion cell consists on two chambers separated by a biological membrane like skin. The donor chamber contains the drug formulation to be tested, and the receptor chamber contains a suitable medium to simulate the biological environment. The skin sample is placed between the donor and the receptor chamber. In the field of in vitro permeation testing, regulatory guidance documents play an important role in ensuring that testing is conducted according to best practices and is consistent with regulatory requirements. Here we have a list of the key regulatory guidance documents used in IVPT studies. This includes the EMA, the USP guideline, OECD, the Scientific Committee on Consumer Safety guideline, and the World Health Organization for dermal absorption.
Regarding human skin samples, it comes from aesthetic surgery from the abdomen area. You can see how skin sample looks like. On this sample, we already removed the fat corresponding to the hypodermis layer, so that we can clearly see the dermis beneath the epidermis. You have to check carefully the skin samples to see if there is any damage or lesion on the skin that may affect the results. From skin sample, we prepare discs of about 3 or 4 centimeters of diameter. As you know, skin thickness can affect dermal absorption. So we need to measure the thickness of each skin sample in order to ensure an homogeneous distribution of the samples within the experiment. At PKDERM lab, we use an electronic caliper to measure thickness of skin samples. That should not exceed 1 millimeter. So this is why we normally use split thickness or dermatomid skin when we perform IVPT. After the filling of a receptor chamber with the receptor fluid, we place a skin sample on the diffusion cell. And this is done in such a way the dermis is in contact with the receptor liquid, while the epidermis is left at the air surface. We then place the diffusion cells on their support under gentle shaking and at 37 degrees. Transepidermal water loss, or TWL, is a quality indicator of skin barrier integrity. So, when performing an IVPT, we need to check the barrier integrity for each skin sample. At PKDERM, we use a theometer system to measure the TWL. Normally, value of TWL should not exceed 15 grams per square meter and per hour, and skin samples having higher values of TWL should not be included in the experiment. For topical application, different forms of finished product can be used in IVPT. For example, we can use a solution, cream, ointment, or gel. For semi-solid formulation, we use a positive displacement pipette for topical application. The amount of formulation we applied on skin surface should be accurately measured. This is why we weight the pipette containing the formulation before and after application on the skin. By this way, we can know the exact amount of the product we applied on the top of the skin. And the amount applied is used to calculate the absorbed dose at the end of the experiment. After the application of the formulation, the diffusion cells are left for the duration of the treatment period, usually 24 hours. For And here you can see how we collect skin sample from the Teledyne Hanson diffusion cell. So you can see clearly the treated area of the skin where we can see the applied formulation on the top of skin surface. And now I'm going to show you how we can remove excess formulation. And this is really the most important step to do in order to get very accurate results. So for that, we use 5 cotton swabs to remove excess formulation. and then we use 2 tape strips in order to be sure we completely remove the excess formulation. After removing excess formulation, we can separate the different skin compartments. We first separate the stratum corneum by tape stripping. So how we perform tape stripping? So as you can see, we press an adhesive tape onto the surface of the skin, and then we remove it abruptly. So this procedure of application and removal of adhesive tape is repeated 10 times. We collect each tape strip in one tube containing an appropriate solvent in order to extract our test article from the adhesive tape. And then we can analyze the compound in each layer of stratum corneum. Normally, one adhesive tape can remove one layer of the stratum corneum. The tubes containing the stratum corneum are then mixed very well and left to under checking for at least 4 hours in order to extract the test compound from the stratum corneum before analysis. After removing the stratum corneum, we can collect the epidermis. For that, we use scalpel blade to remove the epidermis layer, as you can see. The epidermis is then collected in fast prep lysing matrix tubes from MP Biomedicals. To collect the dermis, we use a biopsy punch in order to collect the treated area of the dermis. And then the biopsy containing the dermis is cut into several small pieces using scissors and tweezers. Pieces of dermis are collected in fast prep lysing matrix tubes containing ceramic grinding spheres. These spheres helps to grind hard tissues like the dermis and epidermis. After collection of epidermis and dermis samples, we need to homogenize them for better extraction of the test compound. To do that, we use the fast prep instrument in order to grind epidermis and dermis samples. And at the end, we centrifuge the samples and the supernatant containing normally the test compound is collected. So at the end of the experiment, we get several samples. Samples corresponding to the receptor liquid, samples from the stratum corneum, from epidermis, and the dermis. The samples are stored at minus 20 degrees Celsius before being analyzed.
So IVPT samples need to be analyzed to determine drug permeation and assess formulation performance. One common analytical method used for IVPT sample analysis is LC-MS-MS. So why LC-MS-MS? In fact, because it is a highly sensitive and specific analytical method that can detect low levels of drug molecules in samples. The limit of quantification for analysis of IVPT samples is typically very low, almost 1 nanogram per ml, making LC-MS-MS ideal for detecting even very small amounts of drug molecules that may have penetrated the skin. So high sensitivity is critical for accurate and reliable measurement of drug permeation in IVPT studies. Other analytical methods may also be used for IVPT sample analysis, but LC-MS-MS is often preferred due to its high sensitivity and selectivity. So in conclusion, we saw that IVPT is a critical tool for assessing drug permeation and formulation performance. To obtain accurate and reliable IVPT data, proper sample preparation and analysis are essential. This includes quality control of skin samples, careful application of formulation, removal of excess formulation, and separation of each skin compartment. So IVPT studies are useful in bioequivalence studies, helping to establish the bioequivalence of generic formulations to their branded counterparts. Additionally, IVPT can help guide formulation development and optimize drug delivery, ultimately improving patient outcomes. So overall, IVPT is a valuable tool for understanding drug permeation and has important implications for drug development and patient care.
Thank you so much, Hannah, for this excellent presentation. I'm very confident that our audience found it informative. So we will now move on to the Q&A session. And I would like to remind everyone that you can submit your questions using the Q&A button located at the bottom of your screen.
So thank you very much for the audience, and I would like to thank my amazing team for their big job doing this presentation and performing IVPT studies in the lab. Thank you very much.